who's from Bill DeGrado's lab. I'm sure everybody online is very familiar with Bill's work. Clearly one of the pioneers of de novo protein design, and also lots of other areas as well. And so, yeah, do you want to just take it away then, Nick?
Great. Yeah, so thanks a lot for the invitation to speak. Yeah, I'm really excited about all the potential questions I might get from this audience. So I'm looking forward to that. So what I'm going to talk about for the next 15 minutes is a topic that's near and dear to my heart called the design of small molecule binding proteins. And just this is some work that I've done in the past few years in Bill DeGrado's group at UCSF, funded by the NIH, NSF. So it's a basic fundamental research problem, but it has lots of applications and implications as well for human health. So also funded by the NIH. So I want to start out really with a question, which is why even focus on this? Why focus on the design of binding proteins at all? And I would argue that molecular recognition is really a hub through which we can access all kinds of protein function, right? Signaling, allosteric catalysis, and more, many, many more. Every single protein function, almost every single protein function essentially works through some level of molecular recognition. And so I would argue that if we can't master the manipulation of how proteins recognize ligands, then we can't really begin to design these new kinds of functions from scratch or even understand them. So that's the role that I've focused on in the past few years, is how do we really encode molecular recognition in proteins? How do proteins do that? And can we do that in a computer? And especially, can we do that without relying on just random immunogenesis and screening experimentally? Can we actually encode all these different principles in a computer and come up with a protein binder for a target? So just to remind everybody here, since it's such a diverse audience, what the traditional goal of protein design is it was really to map a structure to a protein sequence. So you start with some target protein structure. It could be actual coordinates, or it could be just a general fold. And the goal was to figure out what sequence or sequences would actually fold to that structure to stabilize it. And we've been remarkably successful in the community over the past few decades, actually. I'm showing top 7 here, Brian Coleman's top 7 and David Baker's group at the top. And I'm showing on the bottom a work from the DeGrado lab, actually, recent work on a membrane protein, helical transmembrane protein by Marko Mravik in our group. And so we can really do this traditional goal of structure to sequence for both soluble and membrane proteins. So the design of a binding protein is a little bit different, though, because there are more degrees of freedom you have to consider. One is you need a target ligand, right? In this case, I'll talk about apixaban in this talk, which is just it's an antithrombotic drug. And so you need a ligand conformation. Which conformation of the ligand are you interested in binding? And which backbone, right? Which protein backbone is actually going to be capable of forming the interactions with this drug, with this molecule? And so there's a lot of unknowns. And one of the biggest questions is, starting from a backbone with protein design, is how do we answer that question? Is this backbone capable of binding this particular ligand? And so there's so many degrees of freedom here. And it's really led to a lot of consternation in this field, essentially just a reliance on random screening and mutagenesis to try to get the job done, and just because there's simply many, many degrees of freedom. And so what I think about, what we think about in the DeGrado group right now, is that the ligand, the small molecule, I mean, it could be any ligand at all. It doesn't even have to be a small molecule. It could be a peptide, or it could be a protein even. It could be a molecular surface. But it's really just a collection of chemical groups, right? And so for apixaban, I'll focus in this talk on these polar groups, because these are really hard to design interactions for, because if you want to bury this molecule for high affinity, you have to dehydrate these polar groups and really form good hydrogen bonding interactions. So these are really the most challenging kinds of interactions to design within protein design algorithms. And so I focus on these, but everything I talk about today really applies to any chemical group as well as apolar. So I'm breaking this ligand into 3 groups, but there really could be more, including apolar groups. And so this idea that this ligand is a collection of chemical groups, it essentially lends itself to a strategy, a design strategy that I've developed over the past few years, and it's been remarkably successful. And so I just thought of an analogy of what this strategy is a couple days ago, and I'm testing it out on this community right now. So it may work, it may not. But the protein design strategy analogy here is made me think about this scene from Willy Wonka and the Chocolate Factory with Gene Wilder, where Mike TV wants to get beamed across the room into this smaller TV. And so if we think of Mike TV here, who's this character here, as this ligand that I want to bind, then what the strategy is to break the ligand into pieces. All right, this is Mike TV floating over across the room, broken into pieces. And I use those pieces, I look for those pieces in the protein data bank. And I learn how proteins interact with the pieces of the ligand. And I can create new structural units that are sort of analogous to rotamers for how proteins interact with those pieces. I call those vandamers. I'll talk about that in a bit. And so once I have these new structural units for how proteins in the PDB interact with the pieces, the protein design process is really how do I reassemble those kinds of pieces within the context or framework of a protein backbone. Here, I'm just going to say this little TV is the protein backbone. And I need to reassemble those pieces within the context of this new backbone and see if that backbone can really support binding the ligand. And so that whole process of reassembly and using these different pieces that I've developed of protein structure, that's a strategy that I developed called COMBS, which stands for convergent binding motifs for binding. So that's the idea. That's the general strategy of what I'm doing. And so in particular, let's just start with how do proteins really interact with fragments, right? And what parts of that do I focus on? So if we look in the PDB, I'm just showing an example of how proteins like arginine, for example, how arginine residues would interact with a fragment like carboxamide. And I have a carboxamide in my ligand apixaban, so this is relevant to my protein design. And so the question is, can I use the PDB to find preferred positions for these chemical groups, in this case, the purple carboxamides? And so it turns out that there are very many preferred, actually very few preferred positions of where an arginine residue would like to place a carboxamide fragment relative to its backbone. Here in green is just the arginine backbone. And so if I turn on the arginine side chains, these are the actual atomic interactions that are being formed with these chemical groups. And you can see that the side chains they can be anywhere really for arginine. There's many degrees of freedom, many rotation, dihedral, torsional degrees of freedom for arginine. And so you can imagine it's just all over the place, and it is. But if we really focus back on where the backbone is and where the chemical group is, and don't worry about where the side chain is right away, we can really start to focus the problem down, essentially having less degrees of freedom to worry about and focusing on the binding function. Essentially, can this backbone place a carboxamide in a position, as well as all the other chemical groups of my ligand, in such a way that my ligand will fit or dock into that, so that that backbone can really accommodate that full ligand. That's the whole strategy. And so just to formalize it a little bit, this idea of what I'm calling a vandamer, it means an amino acid that's in van der Waals contact with a functional group, a chemical group. That is very analogous to a rotamer. And so the idea with a rotamer is that if I have atomic coordinates of my backbone, I know, if given a rotamer, I know where the coordinates of the side chain are. Everything's covalently attached too, right? In a vandamer, we're not talking about covalent attachment. They're non-covalent interactions we're talking about. But it's the same idea, is that if I have a backbone coordinates of a protein, I want to know where's the most probable place to put a chemical group of interest in my ligand. And that's what I developed Vandemers for. I have these for all different kinds of chemical groups. And the idea is to use these then to figure out where these fragments go relative to a backbone. And then if I can piece them all back together to form a binding site for a ligand. And so that's what I do for a pixaban. Pixaban, again, I break it apart into these 3 different fragments, carboxamide, carbonyl. And I was interested in designing a protein completely from scratch. And so using a protein backbone that was de novo, essentially just derived from an equation, parametric equation, using coiled coils. You can do that with coiled coils. So I focused on coiled coils. And so I took backbones like 4-helix bundles that I created on a computer, and I started sampling these Vandemers. What that means is, like for instance, here's a histidine, and here's where a histidine could interact with a carbonyl, and here's a threonine interacting with a carboxamide. And so I start to build up these areas of density of where these chemical groups would like to be relative to the backbone. And what I do with this Combs algorithm is I figure out where the ligand will go, ligand here in purple, such that its groups overlap really heavily and really tightly with all of these chemical groups. You can see it's a bunch of carbonyls here, a bunch of carbonyls, and a bunch of carboxamides. And these are all the interactions derived from Vandemers that come with that. And so I have this potential binding site that I get from this algorithm. And I then choose which residues I want among that set. And the way I choose that is just to maximize the prevalence in the PDB, which ones are used by proteins the most. So now I have a binding site, I have a position of my ligand, I have the interactions, I have the first shell interactions with the ligand, and I just go through a flexible backbone design algorithm to just pack the whole rest of the protein, right? And there's more details in there, but I can't really get into it. And so that's the idea. Order the gene, make the protein in E. coli, and see if it works to bind the ligand, and it does. So this bound the ligand. This is just a binding titration showing that the KD for this particular protein was 5 micromolar. And I got the structure, which is a 1.3 angstrom crystal structure of this protein, and it matched almost exactly with the designed model. So in gray is the design, and then in orange is the actual crystal structure. So you can see the overview here, the density, and so you can really see every atom, except for hydrogens, really. You can almost see hydrogens. And then this is how the binding site looks, design versus the actual crystal structure. So very, very close. I did other binding experiments, and perhaps unsurprisingly, the binding is very, very specific for the target molecule, pixaban. Even if I change it by essentially one group here, I change this carboxamide group to a carboxylate, the KD goes from around 5 to 7 micromolar to 50. If I try to bind a very similar ligand that binds to the same protein that a pixaban does, factor 10A, there's essentially no binding. So it's very specific, and it was essentially identical to the computational model. So this was very encouraging to us. And so really just to recap, since I only have 15 minutes, there are many more details to talk about why I think this worked. But just to recap what the whole strategy this is really just to break the ligand into pieces, use the whole PDB to learn about how proteins interact with those pieces. And then that allows you to answer these questions very efficiently of given candidate backbones, can those backbones really support the binding function? And so really you can go from here, a target ligand, to actually a protein in a computer, make that protein, and it works to bind specifically the target ligand. And I do that using these kinds of tools that I've built over the past few years, Vandemers, this Combs algorithm, this core principle, which I haven't talked about, but you can find in this paper. And so we really are very excited about this, and I want to start to move, not only keep testing this on more proteins that bind ligands for molecular recognition, but start to move this towards even more complex function like allostery and catalysis and signaling. So that's where I'll be focusing in the near future. All right, so thank you.
Okay, thanks very much, Nick, for a fascinating talk, really interesting. So I think Anya is gonna, or Andrew are gonna like sort of liaise and coordinate the questions on our behalf. So I don't know if we have any questions yet, Anya.
Yeah, so we have quite a few questions online. So maybe gonna start from the first one from Don Hilbert. And Don, if that's okay, I'm gonna try to unmute you, and maybe you can ask your question live, please.
Okay, thanks. Great talk, Nick. Can you apply your approach to flexible ligands?
Yeah, so that's really one of the things that I'm working on right now is a different kind of sampling algorithm for allowing flexibility. Because right now, you're right, what I'm doing is I'm targeting a particular conformation of a ligand in the design process. And so for the work that's been published, in order to provide the kinds of alternate conformations of a ligand that could be possible, you really have to just enumerate them beforehand and then go through the algorithm with that static conformation. But there are algorithms that I'm working on right now, actually, that just on the fly allows for the ligand to conform to within the confines of, let's say, a force field to fit into these areas of density that are provided by these Vandermeer chemical group fragments. So I am working on it, yeah.
Okay, thanks.
Mm-hmm.
Okay, great. And we have another question. What are the backbone scaffolds that can support such Vandermeer's placement? Only coils or other folds too? Could you comment on that, please?
Yeah, great question. So any protein backbone this will work for. I focused here on coiled coils simply because I could just create the backbones from the Crick equations. But you could take any protein backbone, your favorite one from the PDB, you could just generate one on a computer. Vandermeer's work on any backbone. And what they do is they allow you to answer the question, does that backbone, it streamlines the answer of the question, does the backbone support, is it capable of forming the interactions, the collective interactions that are required for recognition of a target ligand. But yeah, it works on any backbone. I've benchmarked it on lots of natural proteins like streptavidin with biotin, or paraplasmic binding proteins like glutamine binding protein and its binding to glutamine, those kinds of things. It works for any backbone. It's a very general unit of structure, just like rotamers work for any backbone, Vandermeer's do.
Okay, and the next question from Vitor. How well does the algorithm cope with flexible linkers?
Right, yeah, so it's very similar to Don's question. And I guess to save on time, I could just refer to Don's, the answer I gave to Don, which is that right now the algorithm works on static conformations of ligands, but I'm working on incorporating flexibility there.
Okay, perfect, thanks. And the last question from Amanda Jarvis. Amanda, would you like to ask your question live, please?
Yes, certainly. Thanks for the talk. I was just wondering, can you extend this method to metal complexes and metal cofactors?
Oh yes, yeah. So actually, one of the first tests of this kind of algorithm was for structure determination of a zinc metallic amyloid that we published in PNAS a few years ago with Mei Hong from MIT. And so I used this idea the term Vandemer wasn't invented yet, but the ideas were in our heads. And I searched the PDB for all the histidines that coordinate zinc, and made essentially sets of Vandemers of histidine interacting with zinc. And I used that to help whittle down the possible structures of this zinc metallic amyloid that were consistent with the NMR constraints. And so it really helped us determine uniquely what the structure was, as predicated on how proteins in the PDB use histidine to coordinate zinc. So absolutely, I think metal binding sites, it's one of the easiest things. I mean, I hesitate to say easiest things, but conceptually easiest things, because it's only one atom to deal with, as opposed to I don't have to really worry about breaking that ligand up into many pieces and then reassembling them. The ligand is just a single atom for a metal binding site, or a collection of atoms. And so, so yes, the answer to your question is absolutely, it can be used for metal binding sites, and we've actually been doing that, and I've done that with hemes as well, focusing on coordination of the iron too.